Right. Hi, everyone. I'm Alanna. I am kind of a new PhD. I started in August at Wageningen University, where I'm supervised by Dr. Slayman Schoustra, Dr. Eddie Smid, and Dr. Bas Devan. And today I will present my research proposal, Connected Effects of Diversity and Evolution on Community Function. That's more because I don't have a lot of data quite yet. So to start off, I would like to first point out that I am just one component of a large interdisciplinary project that is a team of PhD students researching entrepreneurship, product development, nutrition, and like myself, microbial ecology in traditional fermented foods from Zimbabwe, Zambia, and Benin. And specifically, my research uses mbisi, it's a fermented milk beverage from Zambia, as a model system for experimental evolution, but you will hear more in just a moment. So as for my talk's title, you'll see that I'm interested in the relationship between diversity and ecosystem function. You can turn this termed as a BEF relationship. And this has been a real focus of ecology with a real rich history of research. And past and current research, there's a lot that uses natural or semi-natural plant communities, as well as microbial laboratory experiments. Although microbial experiments up until recently have largely focused on specific interactions in just one or a few number of species. More recently, microbial studies are including more diverse and natural communities collected from the environment, and also combining these with modeling and from computer mathematical modeling. But there are still components of the relationship between diversity and ecosystem function that are underdeveloped. And most notably is understanding how these change over longer timescales, considering evolution over hundreds of generations. But as we look towards trying to design these longer term studies, it shouldn't come at the cost of losing natural conditions. And this includes assessing and monitoring full communities and their responses, not just looking at select players. And for this reason, I think that mbisi provides a really innovative approach to striking the balance between the control of laboratory studies with quick generation times, with the complexity and resemblance to natural communities. So mbisi is a traditionally fermented milk beverage from Zambia. And unlike supermarket yogurts, it has this complex microbial community, 6 to 10 different dominant lactic or acetic acid bacteria, but also yeast and viruses. And importantly, it's the milk environment. It's easily reproduced in the lab, but still providing a complex substrate with protein, sugars, and even an oxygen gradient due to not stirring it. And in traditional production, there's something called an active backslop method, where a small volume is used to re-inoculate the next batch of raw milk. And this is continued over successive cycles. And it's very highly analogous to the experimental evolution studies that we as evolutionary biologists or ecologists are more familiar with. And lastly, by using and studying mbisi, it's able to combine and look at advanced research across fields beyond ecology and evolutionary biology questions. So I plan to use mbisi communities to study the very broad question of how diversity and evolution influence community function. So that brings to the question of what is a well-functioning ecosystem? Descriptions also often refer to efficient nutrient conversion contributing to growth. So in plant communities, this is estimated as total biomass or productivity. But in microbial communities of fermented foods, it's more suitable to focus on metabolic output and product qualities relevant for consumption. So the acidity, the pH, thickness, and the aroma, the metabolic profile. And especially important are the metabolic profiles, because this indicates at the community level the conversion of nutrients into metabolites. And lastly, another component of a functioning ecosystem is its stability. And this includes responses to newly introduced species, so such as the human pathogen in fermented foods. Now, in evolutionary biology, there's a focus on genotypes and how do these change within a species relating to their fitness, and how well the different genotypes within a species adapted to an environment. Whereas in community ecology, maybe look at a hierarchy, a level above. How do populations of multiple species in a community change in response to selection? Maybe one outcompetes the other, their composition changes. And then perhaps also looking at how does that influence ecosystem function? There seems to be some similarities between the concepts, but are they translatable? And some do believe so. So this brings me to two of my first major motivations and inspirations as I started to think about my PhD project. And the first is the reviews of Mark Velland. So he argues that the framework of population genetics to explain evolutionary processes, this could also be applied to community ecology. So that means viewing different species in a community the same as different genotypes within a species. And this made me think of some of previous considerations by a previous PhD working on Mabisi microbial communities, who referred to almost like community selection, because she saw that compositions of the species compositions changed, but the metabolic profile, the product characteristics of Mabisi remained stable. And the second motivation is work of Andrew Hendry, who points out that studies typically show that a change in ecology leads to a change in evolutionary response, or vice versa. But there's looking at changes inducing changes. We suggest that perhaps evolution can actually contribute to ecological stability, resulting in cryptic dynamics. And so considering this, it means that you do need to look at and assess entire community responses to selection pressures and not just individual players. So I'll explain in this graphic a key framework in my project, and that is the interconnected processes of species sorting, so changes in community composition from evolution, genetic changes within the species, and then how those influence ecosystem function. So you can imagine you have a community, 3 species in their habitat. This is their box there. Each size of the circle represents its population size. In the first case, selection means that species composition changes, but there are no genetic changes. In the second case, species composition remains the same, but there are genetic changes within the species, a lighter color and as a new genotype. In reality, it's a combination of the two. But then you want to consider with these situations, they may or may not have an effect on the function of the ecosystem. So in Mabisi, for example, it's aroma, it's pH. So if there is a functional change, what is the driver? But it's also very interesting if there isn't a functional change. There's changes in the composition of species or genotypes, but the functionality is still stable. So considering this big picture idea, in my PhD, I will use Mabisi to focus on the following objectives. The first is, how does community diversity influence product functionality in Mabisi? Second, how does diversity influence community stability? This will be tested by introducing a novel species. I'm thinking to use Salmonella, a foodborne pathogen. Third, how does evolution over hundreds of generations change ecosystem stability? And fourth, how does starting diversity influence the repeatability, the parallelism in selection trajectories over long-term propagation? And lastly, I won't explain today, but I'd like to validate some lab conclusions with a field study in Zambia. We're using traditional processing methods and the variations involved with that. So my main experiment will consist of creating communities of different diversity. This will be done by diluting a full Mabisi sample, and then they are grown to carrying capacity, so they have similar cell densities, just different diversities. That will be considered the time-zero communities. So 6 of these levels will be then used to inoculate and ferment milk for at least over 250 generations. And where at every fermentation cycle, a sample, a small volume is used to re-inoculate the next batch. And then at different time points in the selection path, I will test the following. Firstly, the DNA for the species composition, the mRNA, which will indicate towards the functionality, the processes occurring across the community, as well as the metabolic profile done through GCMS, gas chromatography mass spectrometry. And then I will look at the stability of the community through introduction of a novel species. I'm thinking to use Salmonella. And then I will also, with these introducing Salmonella, look at how the functionality changes following its introduction. So as through this experiment, diversity will be measured through 16S RNA and looking at the Shannon index of operational taxonomic units. Functionality will be measured through the metabolic profiles through GCMS, also looking at the pH and probably the viscosity, the thickness of the product. Stability will be quantified using the resistance to invasion by Salmonella. So I'll be able to count the number of colony forming units. But I'm also will be measuring how the metabolic profile and possibly the composition changes before and after Salmonella introduction. But any suggestions on how to quantify that per se are very welcome. And then lastly, the repeatability and the convergence across replicates will be analyzed by principal component analysis of metabolic profile and community composition. So with that, a huge thank you again to my supervisors and people who've been helping me out in the lab. And I'm looking forward to hearing your questions, comments, and suggestions.